Friends, this is the second video of microbial pathogenicity. So let's begin. Modes of transmission. Pathogenic organisms can spread from one host to another by variety of mechanisms. First is contact. Contact transmission may be direct or indirect. Direct contact, sexually transmitted diseases, in short STD, such as syphilis, gonorrhea, herpes simplex type 2, and AIDS are acquired by direct contact. Now come to indirect contact. It may be through the agency of fomites, which are inanimate objects such as clothing, toys, etc., which may be contaminated by a pathogen and act as a vehicle for its transmission. Face towels shared by various persons may lead to spread of trachoma. Contagious disease is termed used for the disease acquired by direct contact. Infectious disease is the disease acquired by other modes such as inanimate objects. The distinction between contagious and infectious disease is now not generally employed. Now come to inhalation. Respiratory infections such as common cold, influenza, whooping cough, and tuberculosis are acquired by inhalation. These organisms are shared into the environment by patients in secretions from the nose or throat during sneezing, coughing, and or speaking. Large drops of such secretions fall to the ground and dry there. Small drops, less than 0.1 mm in diameter, evaporate immediately to become minute particles or droplet nuclei, that is 1 to 10 micrometer in diameter, which remain suspended in air for long periods, acting as sources of infection. Now come to ingestion. Intestinal infections like cholera, dysentery, food poisoning, and most of the parasitic infections are acquired by ingestion of food or drink contaminated by pathogens. Foodborne infections occur mostly through carriers engaged in handling or preparation of food and contaminating the foodstuff. The water supply may get contaminated with the feces of or carriers. All these may transmit infection. Now come to inoculation. Pathogens in some instances may be inoculated directly into the tissues of the host. For example, rabies virus is inoculated directly by bite of a rabid animal. Spores of Cl. tetani are present in the soil. They get deposited into the host tissue following severe wounds leading to tetanus. Infection by inoculation may be iatrogenic when on-sterile syringes and equipments are employed. Hepatitis B and human immunodeficiency virus, in short HIV, may be transmitted by use of contaminated syringes and through transfusion of infected blood. Now come to vectors. Vectors are arthropods or other invertebrate hosts. Mosquitoes, flies, fleas, ticks, mites, and lice. Transmission by vector may be either mechanical or biological. Now come to transplacental. Some pathogens can cross the placental barrier and infect the fetus in utero. This is known as vertical transmission. This may lead to abortion, miscarriage, or stillbirth. In case of congenital syphilis, live infants may be born with manifestations of the disease. Infection with rubella virus, especially in the first trimester of pregnancy, may lead to congenital malformations. Such infections are known as teratogenic infections. Other organisms may cause congenital infections are toxoplasma cytomegalovirus, and herpes simplex virus. Now come to iatrogenic and laboratory infections. If meticulous care is asepsis is lacking, certain infections like AIDS and hepatitis B may sometimes be transmitted during procedures such as injections, lumbar puncture, blood transfusion, dialysis, and surgery. These are known as iatrogenic infections. Laboratory personnel handling infections material are particularly at risk. Special care should be taken to prevent laboratory infections. Now come to susceptible host. The infectious agent enters the body by one of the following routes like oral, respiratory, genitourinary, conjunctival, or cutaneous. This is known as portal of entry. Incubation period. It is the time interval between the entry of infective agent and the onset of clinical features of the disease. The infective agent, after reaching the selective tissue, undergoes multiplication during this period. The outcome of an infection depends on interaction between microbial factors which predispose to pathogenicity and various host factors which contribute to resistance. Now come to factors predisposing to microbial pathogenicity. Pathogenicity refers to the ability of a microbial species to produce disease. Virulence is the ability of a strain of microorganisms to cause disease. The pathogenic species M. tuberculosis contains strains of varying degree of virulence as the vaccine strains. The virulence, the virulence of a strain is not constant and may undergo variation. Now come to exaltation. Enhancement of virulence of the strain is known as exaltation. This can be induced by serial passes in an experimental animal. Now come to attenuation. Reduction of virulence of the strain is known as attenuation. This can be achieved by repeated passes through unfavorable host, repeated cultures in artificial media, growth under high temperature or in the presence of weak antiseptics. Now come to determinants of virulence. Now come to under this we will study about adhesion. The initial event in the pathogenesis is the attachment of the bacteria to body surfaces. The attachment of a specific reaction between surface receptors on the epithelial cells and adhesive structures on the surface of bacteria. These adhesive structures are named adhesins. Adhesins usually occur as fimbriae or fibrillae and pili or as colonization factors. Now let's know about the advantages. It prevents the microbes from being flushed away in mucous secretion, urine, and by peristalsis in the gut. Close adhere of the bacterium assures that the toxin will be delivered in high concentrations directly to the host cells. The bacterium is invasive in nature. Adherence helps in penetrating host cells. Now come to besides these adhesions, other structures which play in adherence are outer membrane protein, flagella, lipopolysaccharide, and glycocalyx. Most of the bacteria make use of not just but several of these factors. Loss of adhesions often renders the strain virulent. Adhesins are antigenic in nature. Specific immunization with adhesins has been attempted for prevention of gonorrhea in human beings. Now come to invasiveness. This refers to the ability of an organism to spread within the host tissues after establishing infection. Highly invasive pathogens produce spreading or generalized lesions, while less invasive pathogens cause localized lesions. Some pathogens through though capable of causing fetal diseases, lack invasiveness but remain confined to site of entry and produce the disease by elaborating a potent toxin. Example, tetanus bacillus. Now come to phagocytic factors. Macrophages and polymorphs play a role in phagocytosis of microbes. These are certain factors which oppose phagocytosis or survival of bacteria. These factors are: first is capsule. The cell wall in many bacteria is enclosed by gelatinous layer known as capsule. Most capsules are polysaccharides in nature except that of bacillus which is polypeptide. Capsule contributes to the virulence of bacteria by preventing phagocytosis. Non-capsulated virulent variants usually exhibit low virulence. Now come to streptococcal M protein. The protein present on the surface of group A streptococci binds both fibrin into the bacterial cell wall, is masking the bacterial receptors of the for complement. Now come to cytotoxin. Cytotoxins produced by certain bacteria interfere with chemotoxin or kill the phagocyte. Staphylococcus aureus produces hemolysins and leucocidins. Now come to bacterial surface antigens. The antigen of S. diffi and K antigen of E. coli enable these bacteria to resist phagocytosis and lytic activity of complement. Now come to survival within the phagocytosis site. Ingestion of a microbe by phagocyte results in the formation of a phagolysosome by the fusion of phagosome with the lysosome. Any organism with interference with the formation of phagolysosome can survive intracellularly. Now come to interference with oxidative burst. Superoxide Superoxide and H2O2 produced are lethal to bacteria. Staphylococcus aureus produces the enzyme catalase which breaks down H2O and enables the bacteria to survive. Listeria monocytogenes produces another enzyme, super superoxide dismutase, which neutralizes the oxygen radicals leading to survival of the bacteria. Now come to prevention of fusion and degranulation. That is, cell walls of some organisms modify the phagosomal membrane in such a manner that fusion of lysosome which phagosome is prevented. Example, chlamydia. B. pertussis produces adenylate cyclase which is inhibitor of degranulation and thus enhances intracellular survival. Now come to resistance to lysosomal enzymes. This may be due to the presence of capsular polysaccharides and mycoside. Now come to escape of phagosomes. Some microbes escape from the phagosome into the cytoplasm of the host cell before the fusion of phagosome with the lysosome. Example, rickettsia are believed to escape by the effect of phospholipase A on the phagosomal membrane. Now come to bacterial toxins. Bacteria produce two types of toxins, endotoxins and exotoxins. Let's know about endotoxins. They are lipopolysaccharides in nature and form an integral part of the cell wall of gram-negative bacteria. They are released from the bacterial surface by natural lysis of the bacteria or by disintegration of the cell wall. They are heat stable. The toxicity of endotoxin depends on the lipid component. Lipopolysaccharide binds to neutrophil via lipid A and alter neutrophil function. First point is it decreases chemotactic activity. Second is random migration of neutrophils is decreased. Third point it alters the metabolic and bactericidal properties of the neutrophils. The endotoxins are not destroyed by autoclaving, hence infusion of a sterile solution containing endotoxin can cause serious illness. They cannot be toxoids. They are poor antigens and toxicity is not completely neutralized by the homologous antibodies. Massive gram-negative septicemias may cause the syndrome of endotoxic shock characterized by fever, fever, leukopenia, thrombocytopenia, profound fall of blood pressure, and circulatory collapse leading to death. Now come to exotoxins. They are heat-labile proteins which are secreted by certain species of bacteria. They diffuse readily into the surrounding medium. They are highly potent even in minute amounts and constitute some of the most poisonous substances known. Botulinum toxin is the most poisonous. It has been estimated that 3.0 kg of botulinum toxin can kill all the inhabitants of the world. Exotoxins can be converted into toxoids by treatment with formaldehyde. Toxoids lack toxicity but retain antigenicity and thus induce protective immunity when used as vaccines. Exotoxins are highly antigenic and stimulate formation of antitoxins which neutralizes toxin. They are highly specific for a particular tissue. Example, tetanus toxin for CNS. They have specific pharmacological activities and do not produce fever in host. They are mainly produced by gram-positive bacteria, but may also be produced by certain gram-negative bacteria such as Vibrio cholerae and enterotoxigenic E. coli. Now come to enzyme. Some bacteria produce enzyme which directly damage host tissue. Proteases. First is proteases. These enzymes cleave immunoglobulin IgA which protects at mucosal surfaces. Now come to kinase. It enhances the spread of bacteria by dissolving fibrin clots. Example streptococcus and staphylococcus. Hyaluronidase. It breaks down hyaluronic acid and helps in spread of infection. Example streptococcus. Coagulase. The fourth point is coagulase. It causes deposition of fibrin around the bacteria and thus prevents phagocytosis. Example staphylococcus. Now come to collagenase. It breaks down collagen in connective tissue and contributes in spread of infection. Example Clostridium perfringens. Now come to siderophores and iron acquisition. Bacteria acquire iron for their metabolism. Many bacteria produce low molecular weight compounds called siderophores that can acquire iron from the host iron binding proteins. This property enhances the virulence. Now come to genetic factors. Plasmids are extrachromosomal DNA segments that carry genes for antibiotic resistance known as R factors. These factors are readily transferable by conjugation to the various bacteria and confer resistance to many antibiotics. This also indirectly contributes to in survival of bacteria due to escape from action of antibiotics. Plasmids may also code for enterotoxin production, colonization factors, and siderophore synthesis, and thus enhances the virulence of the organism. All the strains of C. diphtheria produce exotoxin only when they are lysogenized with a bacteriophage called beta phase. The elimination of this phase abolishing the toxigenicity of the bacteria. Now come to infecting dose. Adequate number of bacteria is required for the successful infection. The dose of infects depends on virulence of bacteria. The dosage may be established as the minimum infecting dose or minimum later dose. Minimum number of bacteria required to produce clinical evidence infection in a susceptible animal under standard conditions is called minimal minimum infective dose. In short, MID. MLD is a minimum number of bacteria that produce death of the animal under standard condition. These doses are more correctly estimated as ID50, LD50 as the dose required infect or kill 50% of the animal tested under standard conditions. Now come to route of infection. Certain bacteria are infective when introduced through optimal route. For example, cholera vibrio can produce lesion only when administered by oral route by as unable to cause infection when introduced subcutaneously. However, Staphylococcus aureus can cause lesion whatever may be the portion of entry. Now come to communicability. The ability of a microbe to spread from one host to another is known as communicability. This property determines the survival and distribution of a microbe in a community and not necessarily produce a disease in an individual host. The correlation need not exist between virulence and communicability. Infections in which the pathogen is shed in secretions such as in respiratory or intestinal diseases are highly communicable. In other instances, as in hydrophobia, human infection represents a dead end. Even a highly virulent microbe may not exhibit a high degree of communicability due to its rapidly lethal effect on host. Occurrence of epidemic and pandemic diseases requires that the pathogen should possess high degree of virulence and communicability. Now come to types of infection disease. Infectious disease may be localized or generalized. First is localized. There is a acute inflammable reaction at the site of invasion to arrest infection. When this process falls, the organism spread via the lymph canals or to regional lymph nodes and then to bloodstream, which leads to dissemination of the organism. Now come to generalized. Bacterium, first point comes is bacteriemia. It is the circulation of bacteria in blood. Transient bacteremia is frequent even in healthy persons and may occur during chewing, brushing of teeth, and staining at stools. The bacteria are immediately killed by phagocytes and are unable to initiate infection. Bacteremia of a long duration is seen during generalized infections as in typhoid fever. Now come to second point, septicemia. It is a condition where bacteria circulate and multiply in the blood from toxic products and cause high fever. Now come to third point, that is pyemia. It is condition where pyogenic bacteria produce septicemia with multiple abscesses in internal organs. Example: liver, spleen and kidney. Depending on the spread of infectious disease in community, they may be classified as epidemic and pandemic. Now come to endemic. The disease which is constantly present in a particular area. Example: enteric fever. Second is epidemic, the disease that spreads rapidly involving many persons in a particular area at the same time is called epidemic disease. Example: meningococcal meningitis. Now come to pandemic. It is an endemic that spreads through many areas of the world involving very large number of persons within a short period. Example: cholera, influenza and enteroviral conjunctivitis. Now, finally completed microbial pathogenicity. Any questions regarding this, you can ask me in the comment box and subscribe my channel to know more updates. Thank you.